I'm Chris Whitewoods, and I'm a research scientist at the Sainsbury Lab in Cambridge. So I run my own research group where I look at how leaves are patterned. So a lot of the time we look at how the inside of leaves are patterned. We look at how the air spaces inside leaves are formed and how they're positioned in precise ways to make photosynthesis happen most efficiently. Really, I'm interested in how plants pattern themselves in 3 dimensions, right? So that can be in multiple different ways. So I'm interested in leaves. So that can be how a leaf is shaped overall. So from a flat leaf to a carnivorous plant trap where the leaf is kind of curled up round into a cup shape. Or that can be looking really inside the leaf. At the air spaces inside and how those cells are positioned in 3 dimensions, how those cells separate and make spaces between them, which are then, although they look like nothing, they're actually really important for gas exchange, for photosynthesis and respiration. I went to University in Edinburgh, and when I got there I was interested in kind of biology broadly, so I didn't have a particular interest in plants, and I had a really excellent lecturer called Richard Milne, who he gave some really nice lectures about plant evolution. So the big scale thing about how basically algae dragged themselves out of the water and moved onto land and evolved into all the trees and things that we see around us today. And I'd never thought about plant evolution before, and that really sparked that interest in me. So from there I ended up going and doing a PhD on plant evolution to look at how plants manage to pattern themselves to be able to grow in 3 dimensions, to get enough kind of structure to build a stem that stands up on its own. So I did my PhD in Cambridge, working on a moss, right? To look at how cell division planes switch to pattern a thick stem that can stand up. And then from there, after my PhD, I moved across, 50, 60 miles away to Norwich at the John Innes Centre, where there's a kind of broad plant science community, and I am still working on the question of how plants pattern themselves in 3 dimensions, but instead of working on moss, I worked on a carnivorous plant, and that was really asking the question of how leaves can make such different shapes. So from making a flat leaf, how can they make a actually really complicated cup-shaped trap? And then from there I moved back to Cambridge to start my own lab, and this is where now we're working on the internal patterning of leaves, but still using that same system, this carnivorous plant, which is also aquatic. So it has a lot of kind of strange quirks about it that mean that we can answer questions that other people find it more difficult to. 2 really rewarding things about being a scientist is firstly the research itself, right? A lot of the time research is really boring. I'm sat at a microscope or growing plants, really repetitive stuff. But there's always that moment where, and it's not necessarily the moment you work out how something works for me, it's that moment where you realise that something is strange. So I remember in my PhD we were making mutants. We were turning off genes, breaking genes and looking at what the plants looked like. And we found a mutant where the stem didn't grow. And instead of making a stem, it made this little ball of cells. And at that moment, we realised that this gene that we were knocking out did something really important, but we didn't know how. And they're the moments that are the really rewarding things where you get so excited and realise, yes, this is going to be amazing. And the other really rewarding thing, especially now I'm running my own lab, is it's working with other people and their enthusiasm, especially PhD students and the younger people in my lab, because seeing them kind of get an interest and then develop it and eventually get enough independence to do that on their own, that's incredibly satisfying, and it's almost more satisfying than the research itself, seeing those people take it and run in a direction that I would never have done. So you'll see a research project which you thought that you knew where it was going to go in a completely different direction because of someone else's mind. And that's a beautiful thing.